KURINJI Senior Secondary School, CBSE, Namakkal.
Successful result producing school in state every year.
Administered by the Board of Directors with over 35 years of experience in the teaching field.
Special attention on art, craft, music, dance, silambam, yoga, skating and karate.
Focus on Hindi Sabha and Spell Bee Olympiad exams.
Smart class for effective and innovative learning and well-furnished library.
Solitary importance on sports and games in state and national level.
WhatsApp facilities to know activities of students and school inside the campus.
NEET, JEE, IIT coaching experts from Rajasthan and Andhra Pradesh along with our effective teachers.
Admission registration for classes LKG to +2.
KURINJI Educational Institution, Kavetipatti, Namakkal. Admission in progress. CBSE, KG to 12, contact 9025895176. Metric 6 to 12, contact 9344567484. For online admission, log on to www.kurinjischoolnkl.in.
Students, my dear students, so in this particular video, so we are discussing last and final concept in anatomy of flowering plants. After that one, so we will start to discuss introduction of new chapter. Emma, so let us start to discuss. Emma, so last concept we have seen secondary growth in plants. So for the process of secondary growth, so here 1 or 2 minutes let me revise, later I'll enter into the actual concept. So in the secondary growth, there definitely this particular secondary growth takes place due to the process of or due to the activity of cambium. Okay. When this particular cambium is going to, or it is becoming as or become very active, so thereby we will observe the formation of new structures. So we will observe the development of new structures. So in this particular case, actually few points I didn't give you. Why? Because suppose if I give extra information, so you people are giving complaints, sir, extra information, sir, here we cannot study, likewise you people are saying. So that's why here what is the information what is the information is there in a textbook, only I am going to explain that much of information only. So that's why here one important point, let me explain what is that in a secondary growth. So definitely it takes place in two places. One is taking place in a vascular cambium and one more is in corky cambium. So vascular cambium, what is happening now? Once you see there in previous video, we all know what is that U-steel type of vascular bundles are there. So because of the presence of U-steel type, there intravascular cambium and intravascular cambium's development takes place. So later which one? So due to the activity of cambium, what is happening? So vascular cambium, so on both sides of the cambium we will observe the formation or the deposition of new cells. So what are the cells which are situated outside of the vascular cambium? We are calling as a secondary phloem, and what are the tissues which are present within the like vascular cambium, we will be considering as secondary xylem. So likewise in a cambium or in a corky cambium also, so this particular corky cambium development where we have seen in cortical cells. In cortical cells, so definitely, so why? Because when the pressure once upon in a few days back, we have here one example. Suppose if the room capacity is 100 members or 200 members, suppose if you are trying to keep 300 or 400 members means, so definitely those who are standing in nearby the door, so by the pressure of center one, so chance is there to come out, or those who are standing outside or towards the outside, so they will come out very easily, so from the room. So likewise here also what is happening, so due to the activity of a secondary xylem, somewhat pressure is built up. Somewhat pressure is built up towards the outer side. At the time what is happening, so what are the epidermal cells or what are the peripheral tissues are there, all will be burst out, broken out, likewise we seen. So like in this particular case, some kind of specialized cells which are developed from the cortical regions, cortical regions, so definitely get modified and develops into meristematic cells and start to divide and develops the new cells around of it, so thereby that gives the mechanical support to the plant body. So means in this particular case, no, what are the tissues which are, now from here onwards you listen Nana carefully. So in a KURINJI point of view, so what is tissue, or what are the tissues are present outside of the phellogen, we will be calling as a phellum, and what are the tissues which are situated inside, no, we are calling as a phelloderm. So ellame ipo phellogen, phellum and pelloderm all together we are calling as, once you see here we have written here, so that is periderm. Here periderm means is a combination of 3 different types of tissues together we are calling as, what we are calling periderm. Once you say order wise from upside to downside, once you say first one phellum we will find, later phellogen we will find, later pelloderm we will find out. Okay, so means here the phellum we are calling by one more name, cork. Actually here few important points I would like to explain once you see that. So here the cork is there, no, phellum. So these phellum cells are dead cells, superized dead cells. So these cork cells are made up of suberin. Cork cells are made up of suberin, and cork cambium is there, no. Cork cambium, so this particular cork cambium is living cells. The living cells only, no, definitely having of division of nature. Ipo one interesting phenomenon I'll tell you once you see. Ipo inge cork cambium is there, no. When this particular cork cambium is going to be divided, outside a few cells are developed and inside a few number of cells are formed, no. Ipo interesting point anana, inside cells are living and outside cells means what are the cells which are present outside of the cork cambium or dead one, and what are the cells which are present outside of this one are living. இப்போ பாருங்க epidermis here it is there and it is dead cells, and here the cork cambium is there, and here the secondary cortex is there. இப்போ these cells, so definitely for the metabolic activities point of view, for the completion of metabolic activities, definitely which one is required now, definitely depends on the, or requires, simply I'll explain, requires which one, air. Air is nothing but oxygen is required. Why? Because so what are the food materials which are prepared in the like leaves, no, all will be entered into the different living tissues. So there in eukaryotes, if you see there, no, in eukaryotic cells, so definitely mitochondria will be there. In our 10th standard we learn, so mitochondria is there, that one we will be calling as powerhouses of the cell. Did you remember? So thereby what is the food material is present in the mitochondria will be oxidized, simply digested and energy varo. All these cells are living cells, no, so means energy is required. So energy is required means what is the food materials, or what are the food materials are present here, definitely digest or definitely oxidized. How? So definitely suppose if air comes, suppose if a facility is there for the exchange of gases means at the time, so definitely these tissues receives or the takes the food material exchange of gases or receives the oxygen, and definitely involves in the oxidation of food materials. But here melee dead tissue is there at the time, sir, how sir food exchange of gases takes place, or how sir exchange of gases here, how here exchange of gas takes place means, so here somewhat information I have mentioned. Now after completion of this information, now here let me explain what is the information I have written there. Now at certain regions, listen carefully, so at certain regions the phellum cuts off, phellum. So phellum is the cork only, no, phellum. So that phellum cuts off closely arranged parenchymatous cells on the outside instead of cork cells. Once you see there, so here at a certain regions, a phellum cuts off closely arranged parenchymatous cells on the outside instead of cork cells. Okay, so means here somewhat parenchymatous cells are there. So these parenchymatous cells soon ruptures the epidermis. So ruptures the epidermis and forming lens-shaped structure. So lens-shaped structures. Okay, so now here what is happening? So in certain regions of phellum cuts off closely arranged parenchymatous cells on outer side, so instead of cork cells. So here instead of cork cells, remaining cells definitely cuts off very close to the parenchymatous cells. These parenchymatous cells soon ruptures the epidermis, forming lens shaped openings. So lens shaped opening like lens we will be knowing, no? So lens shaped openings called lenticels. Next one, these lenticels permits the exchange of gases between the outer atmosphere and internal tissue of the stem. My dear students, understood? Same, here we have written same structure, only here only is there once you see. This is lenticels, now we learn. Upper epidermis, so here also epidermis is there. Complementary cells are there, no? All these cells are complementary cells. This is corkicamium is there. Here also we have drawn the corkicamia, presence of corkicamia. Next kile. So here these particular cells only we have been taken as a secondary cortex, and these cells only here we have been taken. Means what is the point now we are learning, no? So this kind of arrangement become like this. Why? So due to the pressure, and here so this particular phylogen cuts off. So here the phylogen is there, no? This particular phylogen cuts off. Here you see there corkicamium phylogen that cuts off, and that particular one and arranged parenchymatous cells are present outside instead of cork cells. So thereby here all these cells, no, we will be considering, or we can be taken them as complementary cells. Okay, complementary cells. Next one here, one point, my dear students, once you see phylogen cells cuts off. Okay, so phylogen cells or nothing, sorry so I think so before I have said I said phylogen cells or cork cells, I think so I said, but don't save that point. Corkicamium. So phylogen is nothing but corkicamium. So these corkicamium cells cut off and closely arranged parenchymatous cells on the outer side instead of cork cells. Means when these particular corkicamium split cut off outside somewhat parenchymatous cells formation or deposition takes place. All these cells arranged like how lens shaped cells. That's why these cells we will be taking as lenticels. And here we can see the presence of parenchymatous cells, complementary cells. My dear students, so sometimes they are asking where we can find out the presence of which one where we can see the presence of this kind of lenticels means, or the complementary cells they are asking. So complementary cells we can see in like before, means in a lenticels structure. Okay, then what is the use of these lenticels, sir? Means definitely involves in the exchange of gases. Definitely involves in the exchange of gases between the outer atmosphere and inner tissue. So between the exchange of gases, why? Because my dear students, here I said no, secondary cortex is a living tissue. Definitely air is required or oxygen is required, I said no. So that's why for sending off or for the transport of or for the exchange of gases point of view, so here the lens shaped structures, lenticels are formed. So that's why here, so I have drawn these two diagrams for your explanation point of view. Again, these particular diagrams also don't consider as a burden to learn. Okay, next one, let us enter into the most important concept, that is secondary growth in roots. Which one? Secondary growth, where in roots. Before what we have seen? So we have seen the information secondary growth we have seen in stem. So thereby when secondary growth starts in the stem, no, automatically that width or the girth of the stem is increased. So girth will be increased, I will learn now. Now secondary growth in roots, once anatomy of dicot stem or dicot root, once you remember. Once you remember the structure, dicot root, once we learn tetrarch condition is there, and exarch condition, xylem elements arrangement, exarch condition is present. Likewise, one diagram we will learn now. So there, what is the point we have learned? So here what is the point here? It is there means, once you see here, I think so this particular diagram is appearing. So once you see, this is metaxylem and here it is protoxylem. One more time, you see it. Exactly at spotlight, what is this space? Pitta, very good. So it is metaxylem and it is protoxylem. Chinnadi proto and perchidi meta. Ipu, what is our point? No? During secondary growth, means in normal conditions, once you see, ipu this line we didn't draw, no? Don't see the labeling. This particular line we didn't draw, just what we have drawn, alternate arrangement, means here xylem, phloem, xylem, phloem, xylem, phloem, xylem, sorry, so phloem, xylem, phloem, xylem, phloem, xylem, likewise alternate, means a radial arrangement of vascular bundles. Emma, did you remember? Very good. So radial arrangement of vascular bundles we have seen in a dicot root. Ipu, in a secondary growth point of view, what is the main important point I am going to give you is, once you see the information, in a dicot root, the vascular cambium or the presence of vascular cambium is completely secondary in origin. Amma point you understood, era. So in a dicot root, definitely the vascular cambium is completely secondary in origin. Secondary in origin means definitely in normal structure, means when the particular root not going to the process of secondary growth, at the time we will not see the presence of cambium. Just we will see only in secondary in origin. Okay, ma. Now it originates from the tissue located just below the phloem bundles. Just below the phloem bundles means here, what is the tissue we can find out? If once you see the labeling, here we are finding this one as endodermis. So here we are finding of endodermis. Next one here we are finding of here we are finding of pericycle, you know. So this particular pericycle, okay. So now this particular pericycle, once we learn now, this particular pericycle involves in secondary growth in formation of cambial wall or the cambial ring. Did you remember? Ipu 2 points we learned, my dear students. So once you see pericycle main important function, lateral roots formation. Did you remember? Pericycle involves in the development of lateral roots, and in a secondary growth it involves in the development of cambial ring. So thereby 2 important functions, don't forget, Emma. So in a NEET examinations, I think so 2 or 3 years before, in a NEET examinations they ask this particular question. Okay, pericycle involves in 2 major functions. One is involves in the development of lateral roots, and the lateral roots origin is endogenous, and one more function during a secondary growth. So this particular pericycle involves in ring formation, vascular cambium, or which one formation? Vascular cambium formation point of view, definitely we have to take the roots will take the help of pericycle. So now let me continue. So what is information? So thereby now one point we are learning now. Vascular cambium definitely developed from the pericycle tissue above the protoxylem. So it is just above the protoxylem, likewise protoxylem information they have given, and a continuous waxy ring. Next one, which layer becomes circular? Once you see, like this. So here, ado madri patamboda the madri irki edo or structure madri, but here finally it will be like converted into like circular. Further events once so here one point we would like to learn once, because of the activity of a pericycle, once the cambial wall formation, so up to here only it is new. Okay, means when due to the activity of a pericycle, vascular cambium formation is taking place, no? So from that, so now here it is there, cambial ring is there between the xylem and phloem members, isn't it? So here xylem and here the phloem is there, between them cambium is there. So once the vascular cambium formation is completed, next one, what are the events furtherly takes place? No, all events are very common as we have seen in dicot stem. So that is only I have written once. You see, further events are similar to those already described in dicot stem. So means once the particular cambium, once the cambium formation is completed between the xylem and phloem members, so automatically, so what are the processes all we have seen in the dicot stem? No, all points are very very common even in root also. So my dear students, what is the point you have to remember in the secondary growth of roots? Only formation of vascular cambium from the pericycle you have to remember. Once from the pericycle, when the vascular cambium formation taking place, so remaining all events, further events all are similar. So likewise are like in dicot stem. So this is a smaller information. So we have to discuss actually NEET examination point of view, even diagrammatical explanation also I have to give you. But so later while I are explaining of NEET information, so 2 more classes extra I will take for this explanation. So still a lot of information is there to be discussed, but regular point of view. So it is enough. So this is a small information regarding anatomy of flowering plants. Ipo en information na new chapter. So new chapter, that is new chapter. Chapter name cell the unit of life. New chapter name is cell the unit of life. Ipo en information. Idi completely like introduction information only, no worry. Right. Now all organisms are composed of cells. One more time, all organisms are composed of cells. Correct only, no? So all organisms are composed of cells. Some are composed of a single cell and or. So what are the organisms are composed of a single cell? All such organisms we will be calling as unicellular organisms. Correct only, no? Uni means single, cellular, only one single cell is there in that particular organism means, so that definitely that particular cell we will be taking as unicellular organisms. So thereby, so here some are composed of a single cell and are called unicellular organisms, and while others like us, while remaining members like us composed of many cells are known as multicellular organisms. Multicellular organisms. So means there are unicellular organisms are there as well as multicellular organisms also are there. Unicellular organisms are made up of a single cell and multicellular organisms are composed by many cells, are composed of many cells. Easy only, no? So now what is the point? Unicellular organisms are capable of independent existence and performing the essential functions of life. So இப்போ என்னென்ன பாயிண்ட் யூனிசெல்லுலர் ஆர்கானிசம் இருக்கா, ஒன்று செல் இருக்கு. இப்போ ஸோ in that particular organism what are the functions it requires? So எல்லாமே means all functions definitely it takes place or occurs in that particular single cell. Even though single cell is present in that particular body of the organism, so that particular single cell is capable for to do the all metabolic activities. I hope you understood my intention. Now unicellular organisms are capable of independent existence, so no need to depend on any other particular other organisms which are present beside to them. Next one, performing the essential functions of life. So what are the essential functions are required for the surveillance and for the life? No, ellame or all functions takes place in that particular single cell. Next one, anything less than, idu koncham nalla purinjukoma, koncham try to understand carefully. Anything is less than a complete structure of a cell does not ensure independent living. Now one more time you listen, at a time you cannot understand. One more time, anything less than a complete structure. Now ange one particular structure miss avana, so at the time no chance for what? Independent living. For us how we will be having all organ systems, no? Likewise, even those single celled body, so there also definitely all kind of structures are required. Suppose one miss one, so even ourselves also, even human beings also, we cannot be said as a complete one. Okay, so likewise here also same point. Hence the cell is a fundamental structural and functional unit of all living organisms. So that's why this particular cell we will be considering as the fundamental structural and functional unit of all living organisms. Simple. So that's why I think so we have learned in lower standards. I think so this particular statement is there. Now that particular statement, so cell is a structural and functional unit of living organism. Next one, so from here onwards actual class will be started. Listen carefully. Now Anton von Leeuwenhoek, actually here Anton von Leeuwenhoek is there, no scientist. Anton von Leeuwenhoek first saw and described a live cell or living cell. So for the first time, who observed the living cell first means answer must be Anton von Leeuwenhoek. இப்போ என்ன பாயிண்ட்னா முன்னாடி ஆக்சுவலி okay for you and in a neat I'll tell you. Sorry, what is the information is there in textbook, that only I'll tell you. Right. Next one, Robert Brown later discovered the nucleus. Anton von Leeuwenhoek so first he was described the or he was seen the which one living cell first. Later next information, Robert Brown later discovered the nucleus. Next one, the invention of the microscope. The invention of the microscope and its improvement leading to the embryo leading to the which one electron microscope reveals all the structural details of the cell. So they have given information regarding the invention of microscope. Once the invention of a microscope is started now, so that time onwards our biology studies everything, so studies improvement also is improved or our studies also is increased. So nowadays we are observing like all kind of structural details information in or under electronic microscope. That is some more information they have given. Now so most important information from here onwards start out. Okay, cell theory in 1838. Even years also try to remember. Suppose in a textbook if they give a year, definitely kandipa try to remember. Cell theory cell theory in 1838, Matthews Sleeden. Okay, surname not required, at least Sleeden you try to remember. In 1838, Sleeden, German scientist, German botanist, examined. So here he was examined. So here a large number of plants. Okay, examined a large number of plants and obtained a large number of plants was examined. And here what is the point here is giving? No. So what is the point here giving? All number of plants he was observed or examined and observed that all plants are composed of different kinds of cells. That is so here all plants are composed of different kinds of cells which form the tissues of the plant. It is easy that we can understand. No, nothing is there to explain. What they are saying, no, Matthew Sleeden, German botanist, observed so many number of plants, and what he was observed, all plants are composed of different kinds of cells. Different types of cells are there which form the tissues of the plant. This is why? Because munnadi we see no simple tissues, parenchymatous tissue, cholenchymatous tissue, sclerenchymatous tissue, likewise we see no. So that is only here saying. Once you see there is composed of different kinds of cells which are which form the tissues of the plant. So thereby we can understand that particular point. Next one, at about some time or at about the same time, not sometime, at the same time, Theodore Swan, at the same time only, Theodore Swan, actually not same time, but they have given 1 year difference is there. It is 1838 and it is 1839. So here Swan scientist, British zoologist studied different types of animal cells and reported that all the cells had a thin outer layer called plasma membrane. So here botanist means German scientist Sleden and geologist Swan so observed German botanist explained regarding number of plants or the so many number of or so much of information of the plants. Swan he was observed different types of animal cells and reported that so definitely on outermost delimiting structure of animal cells, so it was considered as or he was given or used to call it as plasma membrane. But here one more point, what is the information here that particular person was given? No, actually once you see here Swan zoologist means he has right to give any information related to the animal cells, isn't it? But here one point, what is the information they have given here? He also concluded Swanada, he also concluded what is that? He also concluded based on his studies plant tissues the presence of cell wall is a unique character of plant cells. Means here actually he is a zoologist, means he has a right to study or giving of a description about the outermost delimiting structure of animal cell. Outermost delimiting structure of animal cell is plasma membrane, but he is coming forward and he is giving explanation regarding outermost layer of a plant cell is cell wall. My dear students, did you save this point? Outermost layer of animal cell is plasma membrane. Likewise, outermost layer of plant cell is cell wall. That information also was given by Swanada, even though he belongs to zoologist. So, okay, so up to here you try to save in your mind. Remaining information let us discuss in next class. So take care and be safe at home. This particular class here introduction I have given, so that time that side very important discussion we have done. So listen carefully. Suppose if you don't know, so one more time you try to watch. You will be being at home only, so try to watch until you will get the information. Thank you and be safe at home.